I have something here which is fake blood. It's from the party store. I think it's an energy drink. Can anyone tell me why this little bag and its label are important? It says be Rh positive. Have you ever heard of the Rh antigen? There's an antigen on our cells, on our blood cells, mainly, which is called the Rh, which stands for rhesus, actually. The rhesus monkey was where blood types were first found. But no mother wants to hear that she's being tested for whether or not she has rhesus, because they'll go home and look it up and they'll go, "What? You're checking me for monkey genes?" Anyway, there it comes in two forms, so we label it positive or negative. That's all. It's either present or it's absent. If a mother is negative, if the mom is negative and she gets pregnant and the dad is positive, she may have a pregnancy, forgive me, I'm hiding this from you so that you won't won't laugh for too long. This is the pregnant lady. Inside here, she has a Rh positive fetus. It's been shown, and you must have heard this or seen this somewhere, that we can now do diagnoses on a fetus by taking the mother's blood. Because something like 1 out of 1 billion cells is a fetal cell. And we can analyze that by putting it through a cell separator. And these things are amazing. They're like an inkjet printer. So they use electrostatics and a number of other things so that only one cell passes through the detector at a time. And if the cell happens to have the antigen, they will have labeled it in some way with a fluorescent dye, for instance. So as it's dripping down one cell at a time, just like your inkjet printer is putting out lots of little tiny dots, it goes through and it will use either a puff of air or something. If it's positive, it will get shot into that container. If it's negative, it goes into that container for the fetal cells. Well, so now we can do a diagnosis to find out if this fetus is positive. If it's Rh positive, some of its cells get into the mother's cells, into the mother's blood, and the mother's blood, the immune system, recognizes that this is not normal. And so it makes antibodies against it. Which means that if a woman has a pregnancy with an Rh positive baby, and she's Rh negative, if she has another pregnancy with an Rh positive baby, it will very likely die early in the pregnancy, because some of the antibodies will get across and attack the fetus or embryo.
Does that one die?
No, not the first one. Nope. I'm not. So, how do we prevent this? We give a woman something called RhoGAM, RHOGAM. If a mother is found to be Rh negative and the father is Rh positive, they'll give her a shot which will prevent her from reacting to the positive Rh factor.
Excuse me, is that when most babies,
abort miscarriage? Most miscarriages occur very early on, and the woman is actually not aware that she was pregnant.
Right.
And they're mostly because of genetic abnormalities. Mm. And so it's absolutely heartbreaking to talk to a woman who's miscarried after the 3rd month or something like that. And it doesn't console them to say, the baby probably aborted because it was not genetically normal. People they can understand that, but they really don't want to hear it. Don't know any other way to put that. Next class, we're going to be talking about DNA. I'm looking for something which has little balls that are painted different colors.
It was on the table.
Yeah, I know. Oh, here.
Yeah, it is.
Here it is. Okay, we'll be talking about DNA. This is one of the bases or nucleotides of DNA. This is thiamine. And it has carbons, hydrogens, nitrogens, and oxygens. And the thing about oxygen is that if it's sitting with a double bond in one direction, it has electrons on this side that can then have an attraction to one of the hydrogens on the other side. So this is the T of the A, T, C, G, which make up our DNA. I also made the ribose sugar off of this and the phosphate off of this, and they all fell apart. So I wasn't. So DNA is made up of a backbone. I can't tell you how many hours I spent trying to put this thing together. Anyway, here are the parts that make a ribose. Okay. You have a T on one side. It's attached to a sugar. Actually, I think it's a 5-sided sugar. That's attached to a phosphate, which has oxygens attached to it. Let's see, it's going to be 2 phosphates. And then there's going to be another sugar. And there's going to be, here, there's going to be an A. On the other side, you're going to get an A and a T. And these are the hydrogen bonds that I just mentioned, where you have something that has an oxygen, and on the other side you have something with a hydrogen, and they create what are called hydrogen bonds. But it's critical because this is what allows DNA to reproduce. Didn't do that very well.
Theoretically, just out of fun, while you study something like that, would you think of better ways to
Have done it?
Yeah.
Yes.
Yeah.
I do, but that's idle.
Yeah.
That's idle curiosity on my part. I don't. Well, I guess I don't take it that seriously in terms of trying to make myself a authority figure on it. Anyway, this is DNA, deoxyribonucleic acid. Okay, so what that means is the sugar is missing an oxygen. Deoxy. The oxy is gone. Well, what about RNA? RNA is ribonucleic acid, and it has the oxy. That's the only difference between the two. Isn't that amazing that one would get chosen for storage and the other get chosen for messengers?
Yeah.
It turns out, because all of these bonds, all of the single bonds can move, things can fold, unfold, wrap, unwrap, they can be attached to by proteins and so on. It turns out that RNA, which they think came first, itself can act as an enzyme with no protein involved. Fascinating. But it has a different flexibility from the DNA. The DNA is much more stable. It is much less likely to be degraded or to form shapes that can't be useful.